adaptation, or whatever you want to call it. So I'd like to have just a brief discussion about the three major body plan types that occur in animals. And there will be a test question on this. Compare and contrast the acoelomate, the pseudocoelomate, and the true coelomate body plan. So just kind of take note of that. So when we're looking at, I'll go ahead and do it in the same sequence they're doing it here because have it as a handout. I would prefer to flip it over. Have the acoelomate on top and the coelomate on the bottom. That's all right. So in the flatworm, we had a situation like this. This is the acoelomate, or acoelome body plan. Acoelomate just means it kind of implies an organism. Acoelome kind of refers to like more of the description of it. So anyway, we have three dermal layers, or three tissue layers. So let me go ahead and just draw them all in first here. Draw that in. Okay, so on the outside, we are looking at ectoderm. This is usually a single cell layer thickness of outer layer of tissue, ectoderm, that protects the outside. And then we have endoderm, which is lining the gut. And then we have our mesoderm, which is mostly muscles, connective tissue, reproductive organs, everything else inside. So the bulk of the organism is essentially mesoderm. The ectoderm and endoderm serve their function for being exposed to the outside and the inside of the gut cavity for their purposes. But the thing to note here is the term acoelome. Coelome refers to a cavity or a body cavity, and the term a kind of implies no. So there's no cavity or no other body cavity besides the gastrovascular cavity. The gut cavity is the only cavity in the organism. So the gut serves for digestion and circulation. And, it works pretty well. I mean, this is still an advancement over sponges that have no gut, right? So it's kind of the idea. It's even an advantage over the jellyfish and hydra, the Nigeria, if they didn't have mesoderm and their body is real flimsy and delicate. So we're having kind of organisms that are more robust, can be larger, have more locomotion because of muscle tissue, etc. All right. Then the next idea we covered last time on the nematodes was this idea of a let me redraw that. Okay. So this is going to be called the pseudocoelome. And we know that coelome means cavity. Pseudo means false. And coelome means cavity. I mean, it is a true cavity, but it's not a fully lined cavity, is the idea. So we have our tissue layers. Go ahead and draw them in using the same colors. Okay, so we have our endoderm. We have our mesoderm. And then we have our ectoderm here. So the same tissue layers, but the difference here is surrounding the gut is this cavity that functions as a circulatory cavity. So when the food is absorbed from the gut, then the nutrients can be transported around by this circulatory cavity, right? And other functions as well. I mean, organ storage, what have you. But we're separating digestion from circulation, which is kind of makes sense, so to speak. So today we're going to cover the annelids. We're going to focus in on the annelids, the segmented worms. And the segmented worms represent the first organism that has what's called a true coelom body plan. And then everything else this semester is going to be a true coelom. That's like the main type of animal body plan. Annelids, mollusks, arthropods, vertebrates, everything else has a true coelom. So it seems to have been very successful, very useful plan. Okay, so we have our ectoderm. We have, let's go ahead and do our endoderm here. All right, and the main difference here is the mesoderm lines both sides of the cavity. On the outside, and lines the gut. So we have mesoderm, okay? And this is called a coelom. This cavity, because it's entirely lined with mesoderm. There's only mesoderm lining it. The big advantage here that implies is that the gut now has mesoderm tissue for organ development. So this is where we can develop things like stomachs and crops and gizzards and all kinds of stomach organs and or gut organs can be developed because of the mesoderm lining around the intestinal tract. This couldn't happen in the coelom body plan. Okay, so that's kind of the theme here for the segmented worms is what happens in their plan, and I'll kind of point out some issues that arise from that. Okay, all right, good. So there'll be a test question that asks you to compare and contrast a coelom body plan, a pseudocoelom body plan, and a coelom body plan. And I would expect you to be able to provide a little diagram like this, a little sketch. It doesn't have to be colored, but we have to demonstrate to understand what a true coelom is versus a pseudocoelom versus an acoelom. All right? Sound good? Okay, so last time we went through the nematode and let's see here. Yeah, we'll look back at the nematode to compare it to the segment. So the segmented worm is our true coelom type organism called Annelida. Annelida. These organisms have a segmented body. Draw kind of a side view here of them. Okay, so there is our segmented body. And then we have our gut, which, because of the mesoderm lining of the gut, we have gut organs, including pharynx, crop, gizzard, hyposole, which is our gut tube. Okay. Then we have mesoderm kind of lining the inside had the body here. Right. And line you get. So, okay. There we go. Okay. So there we got our kind of our annelid body plan. Now, one thing to note is that the segmentation wasn't just external on these earthworms. So this body plan with the coelom cavity in the middle is kind of like the nematode, where we have the ability to use the coelom as a circulatory cavity. They actually had their body was kind of divided into compartments, and the adaptation for dividing into compartments here, internal segmentation, was for locomotion. They used their water pressure, or the hydrostatic pressure, inside the compartments to, if they squeeze their circular muscle, they can extend their body, and then if they retracted their longitudinal muscles, they can shorten their body. So they were using the individual compartments almost like it's called a water skeleton or a hydroskeleton. So they're using their body compartments for locomotion, which is a great advantage, because here's the first organism that has kind of the ability to really have extensors. So we see clamworms being able to reach out ahead of them and grab prey, and that's going to be a major advantage compared to the nematodes, which kind of just bend. I always kind of think of nematodes as being kind of like this ruler. They just basically kind of do like a serpentine motion like this, but they can't really reach out or push forward. But the annelids were able to actually move their body away from the center, which is kind of a new body motion. And then obviously we saw the squids do that. The squids have the same idea. They have a water skeleton also. So, so the water skeleton is a huge advantage in this body plan. But the problem is, if you're using your coelom as a water skeleton with compartments, you really no longer have the ability to use it for circulation, right? So it's like now we have the food is brought in here and kind of mashed up a little bit and separated. It's broken down in the crop and gizzard, and then it's absorbed in the intestines. So nutrients that are absorbed back here in the intestines, basically, how do you get that nutrient back up to the head of the organism? And the problem is these compartments are basically keeping the coelom fluid from moving around. So here is where we have an adaptation of a circulatory system. So these organisms have evolved a type of circulation that enables them to move material around. So here we go. Let's go ahead and label this here. This coelom compartments. Is used for a water skeleton, so it can no longer really be functional as a circulatory system. Let me go ahead and redraw this without all the compartmentalization. Go ahead and redraw a segmented worm just on the outside. Okay, this, and let's go ahead and I'll go ahead and put it in the gut. Alright, so there's basically the same idea, but now what we have going on is we have a circulatory system. It includes a dorsal blood vessel and a ventral blood vessel. And the circulation was something like this. We had 5 aortic arches We got these aortic arches. And the blood fluid basically circulates towards the head dorsally and passes ventrally through the aortic arches, this, and then passes posteriorly here. In the back of the body, there is capillaries that allow the blood to kind of migrate from the ventral side back to the dorsal side. So here we have this, what's called a closed circulatory system, which means that the circulatory system is separated from the coelom system. The circulation is separated from the body compartments. So what this does is this overcomes that the coelom has been adapted for movement by blocking off these compartments, and now we can circulate nutrients, oxygen, wastes, etc. around the body. Isn't that cool? Okay, so that's a major adaptation for a number of things. We could say, okay, so back to our plan, right? Didn't we talk about feeding, respiration, nitrogenous waste disposal, and osmoregulation, right? That's kind of our theme. So now that we have this body plan idea, let's go ahead and cover that. So feeding. The advantage to feeding is with a mesodermally lined gut, we have feeding organs like stomach, or we'll call them a crop and gizzard. But basically it's like feeding organs. Crop, gizzard, and then typhloid. So the main point there, though, is that they have developed the ability to further process, or maybe better process the food than the nematode, which just had like a tube. It was undifferentiated. So for feeding, the advantage is mesodermally lined gut tract improves digestion ability, or opens up a wider variety of food resources. For respiration, these organisms do, I guess what you'd call it cuticular respiration, but essentially it's like simple diffusion. Oxygen diffuses into the body from the surrounding atmosphere or water. Let's go ahead and just say water in this case, because most of these are aquatic or marine. Oxygen diffuses in through the skin by simple diffusion, but the enhancement here is the circulatory system is going to carry that oxygen all throughout the body with a little bit more efficiency. And the closed circulatory system also includes blood kidneys like hemoglobin. Which is a iron-based oxygen carrier. Hemoglobin basically uses iron to carry oxygen, and that enhances respiration. So we have respiratory adaptations of a circulatory system that includes a blood protein that carries oxygen and iron, and that's a big advantage. Okay. Got it? Okay. Then let's see. For nitrogenous waste disposal, the same kind of issue applies here, is that because the body is divided into compartments, you really can't circulate nitrogenous waste through the body, or get rid of it from deep inside the body quickly. So the nitrogenous waste has to be converted into something that can be temporarily stored. So urea or uric acid, usually urea would be the kind of primary waste here. But these organisms have a really important nitrogenous waste disposing structure called an nephridium. And when we dissected these earthworms, I asked you to take a look to see if you could find these, because they're really kind of an important simplified version of a widely used nitrogenous waste and osmoregulation structure. In fact, you have a form of nephridia that's probably homologous to this nephridia, so it's very old, very successful. So let's go ahead and cover that. To do that, I'm going to draw yet another worm here. I'll do a shorter version though, to show the nephridia, because these are getting kind of complicated, with all the stuff going on inside there. We never knew worms were so complicated. Okay, so here we have kind of a abbreviated, shortened version of an earthworm here. And let's go ahead and do here we go. Let's do this. Trying to think how to make this simplified. Okay, I'm just going to go ahead and divide this into compartments like this. So here we have our internal compartmentalization of the gut. And the nephridia are, let's see if I can get this perfect. The nephridia are these little kind of like hairpin loop-like structures that there's a pair of them at every segment. So it looks something like this. It's kind of like a trumpet or a horn or something like that. So let me draw a couple of them at these segments here. Okay. So what they do is the nephridia. Let me do this in all label parts. Parts are important. Okay. So here's 3 nephridia. There's basically a pair of them in every segment, from the head to the tail of the organism. There's a pair of them. All right. So the front part of the nephridia, or the definition of the nephridia. So these are nephridia. And what the nephridia is, it's a ciliated tube that filters the sebum. So this is the ciliated nephrostome. Nephrostome means nephridia mouth. So the cilia will generate a water current and pull water fluid out of the coelom here and pull it back into the nephrotubule. This is the nephrotube. Nephrotubule. And then wastes can be concentrated by either retaining water. Water can be pulled back out of the nephrotubule back into the coelom compartment. Salt ions can selectively be either taken out or concentrated, gotten rid of. And then the wastes are expelled out the nephrodiopore. Nephropore. So the main parts here are a ciliated nephrostome, a ciliated, like a trumpet-like opening. The sebum fluid, including the wastes, water, and salt ions, are drawn in. Wherever the organism lives, if it lives in the marine environment, they want to retain water and get rid of salts. If they live in a freshwater environment, they want to retain salts and get rid of water. If they live in a semi-terrestrial environment, they want to retain water and salts. And so the general idea here is this is a really flexible, really adaptable, very, it enables them to live in a wide variety of habitats. So that's kind of the benefits of a nephridia is it is a very useful adaptation for adapting to a wide variety of habitats. Let's go ahead and do this where we can concentrate. We can concentrate nitrogenous waste like urea. We can do things like retain water. Where's my dark blue here? We can retain water, and salt ions. Salt can be exchanged. And so I just kind of want to really emphasize that this is a very important structure, and we're going to see this structure pretty much in almost all the rest of the animals this semester. So the annelids will have them, mollusks have them, the chordates or vertebrates have them in some capacity. The only organism that doesn't have them is arthropods. And probably the reason arthropods don't have them is arthropods have no ciliated cells. So for some reason, arthropods lack cilia. And so that basically shuts out the ability to have nephridia. But they have developed a different mechanism. It's kind of like nematodes technically. So obviously it didn't hurt them with their diversification. Okay, any questions? So as far as water balance on these organisms, the annelids are all osmoregulators. So in the marine environment, the water is about 3.5% saline. They maintain, somewhere around a 0.9 to 1% saline equivalent, or their osmolarity is about 1/3 of what seawater is. And they're able to maintain that because of these nephridia. And then in freshwater, I mentioned that no organism can be an osmoconformer, so they're able to invade freshwater and be successful. And it probably enabled them to be semi-terrestrial or live in moist soil environments, etc. Now good? Okay, so a lot going on in the annelids. There's a lot of complexity in terms of skeletal features, in terms of circulation, in terms of nitrogenous waste, digestion. So it's a big jump in advancement here. Got it? Okay. So, let's go ahead and move on to the mollusks. And about them. Okay, so when we talked about the mollusks, so the cool thing, that what makes it easy for us, is the mollusks pretty much have all the same kind of advantages that the annelids have. They have a true coelom, so they have a mesodermally lined gut tract. So for feeding in mollusks, they have the same advantages of having digestible organs. For nitrogenous waste disposal, they have a pair of nephridia. So they don't have a pair they don't have multiple pairs. They basically have one pair for the whole body. But the big difference between, and they have a circulatory system of sorts. Basically the ability to circulate blood fluid around. Let's go ahead and draw a, kind of like a snail or something as an example of a mollusk here. So mollusca. So I'll draw a little snail here, kind of idea. Let's see here. This. Better antenna. Okay, so let's see here. For the digestive system, yeah, like this. Give them a little stomach. Usually the gut exits into the mandible cavity like that. So there's our gut. Okay, so the one thing I want to point out about the so far, feeding is the same idea as the antlet. They have a mesodermally lined gut tract. I'm not going to draw that in, so I don't want to get this too complicated. But they have a mesodermally lined gut tract, digestive organs, etc. So for feeding, they essentially have not much going on different than the antlets have. It's a good advantage over everything else we've seen so far. For respiration, there's our big theme. Respiration is the new idea. This is the first organism that had a respiratory organ, and they have this gill called a cnidia. So, I mean, it's commonly called a gill, but for mollusks, it's a unique structure because it is a gill that kind of hangs in their mantle cavity. That gill is associated with a pericardium or heart. Okay, so what happens is their coelom fluid is drawn or pulled up from the coelom by the heart, and then the blood fluid is then circulated through this cnidia and then dumped back into the body. So they have a circulatory system, different from the annelids, but it functions the same way. It circulates blood fluid around the body. So for respiration, the big advantage of the mollusk, that's the big jump there, is they have a respiratory structure, a gill, in order to enhance respiration. In addition to the gill, they have a respiratory pigment called hemocyanin. We'll probably talk about hemocyanin maybe today, certainly on Tuesday. We'll see what we get to. But hemocyanin functions a lot like hemoglobin. It uses copper to carry oxygen. And an interesting little factoid is it takes 2 coppers to carry an oxygen in hemocyanin, whereas it only takes 1 iron in hemoglobin to carry oxygen. So it's not as efficient of a respiratory pigment as hemoglobin under certain conditions. Under other conditions, it's more efficient. So we'll discuss that in a bit. But just to note that mollusks also have a respiratory pigment that's different from what they generally have. All right? So we got feeding, right? Same as the annelids. We got respiration, different from the annelids. They have a gill, the annelids don't. They both have respiratory pigments, but they're a little different. Okay, nitrogenous waste, they do have a nephridia. So I can go into this that they use to filter nitrogenous waste out of the coelom. When they are small, when they're small marine mollusks, like a little baby limpet or something, they can get rid of ammonia. When they're larger, like an abalone, they can get rid of urea. And when they are a terrestrial snail, like a garden snail or banana slug, they can get rid of uric acid. So the mollusks have a wide variety of nitrogenous waste excretion abilities, which probably enabled them to be, this is the first organism that we're talking about that has colonized land. Mollusks were probably the last phylum to colonize land. Arthropods were first, vertebrates were next, the amphibians. If you've been doing your geologic time scale, the arthropods colonized in the Silurian, the vertebrates colonized in the Devonian, and the mollusks colonized probably in the Carboniferous. So it's in that sequence where they each kind of took their turn getting on the land. Okay? Sound good? Okay, so Nephridia enable them to live in a wide variety of habitats, saltwater, freshwater, and land, and they are all osmoregulated. Okay, so I think for the general function of what do they have for feeding, respiration, nitrogenous waste, and water balance, I think that we got the whole story here, right? The big difference between mollusks and annelids is the respiratory organ, right? Okay, so now what I want to do is talk a bit about what adaptations they had, or maybe pre-adaptations, or exaptations they had for moving on the land. And that's actually an interesting story. It's like, okay, so they have these unique mollusk features. Some of those features enabled them to actually transition onto land. One of those things for water balance is they have a shell. The shell, at least in snails, provides protection from not only predators and injury, but it also is a kind of like a house that keeps you from drying out. So they cover up a major part of their body with a protective shell that keeps water handy, right? Keeps water inside. In addition to the shell, they have slime. And so the slime, or the mucus, I guess slime is so judgmental, we'll call it mucus, right? Mucus, we'll go ahead and put slime. So the mucus is basically it has a protein-rich kind of moistening juice that secretes from their body. And that mucus helps prevent them from drying out. If you've ever handled a banana slug or a snail, you know that once you get that slime on you, it's like hard to get off, right? Sticky and kind of goopy and kind of like rubber cement or something, right? And there's a really fun link here if you check out these PowerPoints. Some beauty salons specialize in letting large snails crawl in the face, and that slime actually is the right treatment. And the disease cystic fibrosis, which is a genetic disorder that basically chloride ions build up in your cells. And the problem with that is that chloride ions cause your lung cells to dump water into your lungs. And so people with cystic fibrosis have a serious health condition. They have actually looked at using slime treatments to treat cystic fibrosis disease. So there is some medicinal benefits to this snail and this slug slime. So interesting stuff. Interestingly enough, that you probably know that one way to kill garden slugs and snails is to put salt out on them, right? So what does salt do to the snail? Yeah, well, what you're doing basically is the mucus that normally protects them from drying out or water balance, you make suddenly hypertonic, right? If you put salt in their mucus, the mucus now becomes hypertonic and pulls water out of their body, right? So essentially, you're using their mucus against them. And that's why when you put salt on them, they kind of sizzle and fizzle and all that, all the water is being pulled out of their body. And yeah, it does kill them, but but that's just kind of an interesting thing to note, that the reason why salt kills slugs and snails but doesn't really harm, insects. You can't kill cockroaches with salt. They're like, bring it on, right? But snails and slugs, it kills them because you're basically reversing the function of the slime that protects them in the first place, right? Okay, so that being said, they had a major opportunity to move into terrestrial environments. And one of the issues of living on land, moving to terrestrial environments, is drying out, or what is called desiccation. Oftentimes, I'll have a fun little class activity where I ask the class to spell desiccation correctly, and this is always fun because no one spells it correctly. Or usually, I've never had a class where someone calls out the correct spelling desiccation. Now with smartphones and things like that, it's like no fun. So anyway, drying out, the issue of living on land is called desiccation. And desiccation is spelled like this, with one S and two C's. Desiccation. And what desiccation means is when you live on land, water leaves your body. You have, you're drying out, your cells are losing water to the atmosphere, essentially, desiccation. So we'll revisit this term when we talk about terrestrial respiration for lecture exam 3. But anyway, so let's discuss some of the mechanisms by which mollusks have moved onto land. And the shell and the mucus are adaptations that enable them to move onto land. The final thing they had to do was to get rid of the gill. The cnidia, being a large surface area for respiration, risks losing lots of water to the surrounding atmosphere. So all of the snails and slugs that have colonized land have basically lost their gill. And now what they have is, say for example, like the banana slug example I gave. The banana slugs, our banana slug, something like this, okay? Now what they have is a lung cavity. So this is what is now called a lung, but it's basically the same mantle cavity that the snails have. And then they have an opening to the lung that I call a neustome. Certainly on the lab exam, you're going to have to know the name of that neustome thing, because this basically is like it's an opening to the lung cavity that they can open and close it as needed to help conserve water. So the issue with living on land is desiccation, meaning you lose water to the atmosphere. And how the mollusks has gotten around it is they had already shells of mucus to help them prevent from drying out. But losing their cnidia, losing their gill, is kind of the primary thing that enabled them to move on to land. And we'll cover this more in lecture exam 3, but the atmosphere has about 35 times more oxygen available than the water environment. So because there's so much more oxygen available on land, you don't need to have as big of a respiratory surface area on land in breathing atmospheric oxygen. Okay, I think what we're going to do is I'm going to push out talking about open versus closed circulatory systems until Tuesday. I see what we've done here is we have our lecture exam on the 24th, and that means that I have several days to cover stuff. So I fear that if I rush it, we're going to be like out at 17th with nothing to cover, and wishing that I had the exam that day instead. So we'll go ahead and stop there. Today we have crustaceans is what we're covering in the arthropod world, and that's a fun one. A little crayfish section. So let's go ahead and stop there since we're almost at 10 till anyway. You can get your picture on the board, but think of like the things that, like what kinds of questions would I be asking on the exam about these things? Probably. So how I've done this, just to give you a hint, how I've covered this stuff is I've covered it by phylum. Like, so by phylum, I've gone like, okay, what do the sponges do? What do the Nigeria do? What do the flatworms do? What do the nematodes do? What do the annelids do? What do the mollusks do, right? Like this, I've gone by phylum. On the exam, I'm going to flip it around, and I'm going to go by system. So it'll be like feeding across the board. What are the major jumps or advantages that happen in feeding? So comparing, say for example, sponges to Nigeria, the jellyfish, what was the major difference in feeding? And it was the gastrovascular cavity pocket, right? Like the gut pocket. That's a big advantage, right? But like, I wouldn't say compare Nigeria to flatworms for feeding because they had the same thing, right? Didn't they both have a gut pocket? So maybe it'd be more interesting to go compare sponges versus Nigeria versus like nematodes for feeding, right? And then didn't we have like no gut pocket with the sponges? They had to feed on food smaller than their cells. Gut pocket with the jellyfish, they could feed on larger food items. And then the nematodes had a gut tube, right? Like a gastrointestinal tract. So that's kind of how I'm going to do it on exams. I'll have like a compare and contrast 3 or 4 different organisms by physiological systems. Feeding, respiration or gas exchange, nitrogenous waste elimination, and osmoregulation. So just think about that. It's going to be the same information, but the test will test you on like comparing these physiological systems, even though what I've done in class is I've gone through by phylum. So just kind of think about that, how to do that, how to study for that. But we'll continue on Tuesday with comparing the open versus closed circulation systems. We'll start there. Let's take I got a long 10-minute break. Like 5 minutes after, I have to look at my crayfish and get some crustaceans out, get the cart, and we'll go. Sounds good.